I'm going to take you through a little bit of background, very superficially, about hydrogen sulfide and what it does, and why some early research we did on that gaseous transmitter led us to believe we might be able to exploit that molecule to make safer and perhaps more effective drugs for a number of different conditions, but primarily anti-inflammatory drugs. I'm also going to take you through some of the trials and tribulations of our attempts to translate what we learned to the clinic and eventually to a marketplace. This might precondition you to never want to attempt it yourself. I hope not, but it may be. This is Toronto, which is where our company Anti Therapeutics is based, and then I have my university appointment on the other side of Canada in Calgary, and also increasingly spending a lot of time doing work in Brazil, which is great. So the story starts 600 million years ago, when there was no oxygen, but when we did have life on Earth back here, and there was plenty of hydrogen sulfide around, and those organisms adapted to use hydrogen sulfide as to generate energy, to generate ATP. There was, as I said, go back. And in fact, our mitochondria have retained this capacity to use hydrogen sulfide as to drive energy production, to drive ATP formation, and particularly in circumstances of hypoxia or anoxia, small amounts of H2S can rescue cells. And that's what's diagrammed here. We do have at least three different enzyme systems for generating H2S in mammals. We also have the microbiome, which has the capacity to generate H2S, probably generates a little more than half of what we have in our bodies. And H2S, as I said, can drive ATP production. The best adapted cells in our body for using H2S as an energy source, I don't think it's a coincidence, are the cells that line the gastrointestinal tract. They're very efficient at converting H2S to energy. That's what's shown down here in the bottom. This slide just is a bit busy, but shows some of the main properties or effects of hydrogen sulfide that led us to believe it could be a useful molecule for drug development. And this was the first discoveries we made related to H2S, was that H2S was extremely potent at inhibiting leukocyte adherence to the vascular endothelium and transemigration of the leukocytes across the endothelium, as well as endothelial damage. There's also good evidence that H2S promotes resolution of inflammation. It can induce neutrophil apoptosis. It can drive the change of phenotype of macrophages from M1 to the pro-resolution M2. It inhibits some of the cytokines produced through NF-kappa B and promotes NRF2 activity. And there's many other actions of H2S, all suggesting very beneficial effects at physiological concentrations. And I'm going to talk today mostly about things going on in the GI tract. That's my background, but they pertain to systems throughout the body. And this slide's incomplete, but H2S has been shown to have cytoprotective, pro-repair, anti-inflammatory effects in all organs of the body. So my interest for many years has been on the ability of this class of drugs, non-steroidal anti-inflammatory drugs, or NSAIDs, to produce injury in the gastrointestinal tract, as well as to produce cardiovascular adverse effects. Despite many, many new NSAIDs introduced over the years, despite many other products that are supposed to protect our GI tract from this damage, the problem persists. The problem has not been solved. Many people take NSAIDs on a daily basis. It's the #1 cause of drug-related death in hospitals. There's still significant deaths and also significant hospitalization. So it's a problem. It's still an unmet medical need that we need to solve. And if you look, as we went into this thinking we might be able to design NSAIDs that are better, that are safer, consider this. The clinical use of NSAIDs is substantial. It's about a $6 billion a year market. But the toxicity of NSAIDs, both in the GI tract and cardiovascular primarily, kidney as well, limit the use of NSAIDs. So we have people who can't take NSAIDs and they take drugs like acetaminophen instead, or they take narcotic analgesics. And those two classes of drugs also have significant adverse effects and cause significant healthcare costs. If we could solve the toxicity problem with NSAIDs, we could expand the use of NSAIDs into those other areas with just through increased safety. So that's what we're attempting to do. And just to round up this part, just summarizing about NSAID enteropathy, it is the case that despite efforts over the last 60 years to make better NSAIDs, to make NSAIDs that don't have the adverse problems, it does remain a significant problem. The drugs designed to prevent the major toxicity of NSAIDs do not solve the problem, they move the problem. We're really good at protecting the upper GI tract against damage, and all we're doing is shifting that damage into the small intestine, where it's more difficult to detect, where there are no drugs for prevention, no drugs for treatment, and where the hospital costs associated with that damage is significantly higher than the upper GI damage. So we're just sticking it away in a closet. So, some years back, 2004, I founded this company, Anti-Therapeutics, with a goal of designing H2S releasing NSAIDs that would have a greater safety profile than existing drugs. So we just did a simple approach. Take an NSAID, covalently link a hydrogen sulfide releasing moiety to it, and see what happens. And we did basic research showing that H2S donors, just given with NSAIDs, did have a protective effect. So we took a whole range of different NSAIDs. This is some of them. We took some H2S releasing moieties. I asked chemists, could you stick these two things together for me? And they did that. And the drug that we decided to focus on for a particular reason was we chose naproxen to be our base drug because at the time, and to some extent it's still true, naproxen is recognized as the most cardiovascular safe of the NSAIDs. So we thought we may as well get the most safe on the cardiovascular side to start with. And we chose this particular H2S releasing moiety for our compound, which is 4-hydroxythiobenzoamide, or TBZ. So this is our lead drug, ATB346. To the left of the blue line, that's naproxen, and to the right is this TBZ molecule. Now, a point I want to make now, and I'll come back to it later, and something that we have sometimes forgotten in the history of this company, is that ATB346 is not a prodrug. It's not the case you give the drug, that bond gets split, and naproxen goes and does all the good things. And you're going to see some evidence of that throughout the talk, but here's some right off the bat. If we take some rats and we administer naproxen in blue or ATB346 in red, and we measure plasma naproxen levels, you can see that when you give naproxen, it rapidly rises in plasma, and it, over the course of 6 hours, declines a bit. But ATB346, you can see in these initial hours, there's hardly any naproxen. It's barely detectable. It's certainly not at levels that would produce an anti-inflammatory effect. But if you look at pharmacodynamics, so the inhibition of COX activity, so the enzyme that NSAIDs target, so with PGE2 synthesis as a readout, these are from the same animals. You can see both naproxen and ATB346 in this early time period are producing equivalent suppression. So despite there not being any naproxen in blood, or very little, we're getting a good effect. So either ATB itself or a rapidly formed metabolite is blocking COX very well. So I'm going to take you through some of the kind of key data in a pretty superficial way, just to convince you, as we were convinced from animal studies, that this drug had some promise. And the efficacy data I'll show you wasn't done by us. It was done independently by a contractor in France. And I apologize, but these slides were made for a business audience, so they don't have complicated things like standard error bars that are apparently very confusing. So this is an adjuvant, a rat adjuvant arthritis model, where you give adjuvant, and over the course of a couple of weeks, they develop arthritis, and you can measure the change in paw volume. The paws get thicker. This is the control group in black, so you get a nice inflammatory response over the 2 weeks. If we treat with naproxen, the blue line, at 1 milligram per kilogram or 10 milligram per kilogram, you get a nice dose-dependent reduction of inflammation. You can see in red, that's the equimolar dose of ATB346, producing a similar effect. And there's actually one more line here. The dotted red line is ATB346 at the equimolar dose to 20 milligram per kilogram naproxen. And you can see the blue line stops there because all 12 animals died from intestinal perforation. So it's an efficacy study, but we did get a toxicity readout as well. showing that our compound was safe in these animals at a dose where naproxen was causing significant damage. What about upper GI damage? Well, we've done a lot of studies. I'm just showing you a few today. We've done a lot of studies looking at the ability of NSAIDs, naproxen and other NSAIDs plus our drugs to produce gastric damage. When you give an NSAID to a rat that's been fasted overnight, this is the kind of damage you see. These are hemorrhagic lesions. Anywhere you see dark red or black, it's blood, stained black by acid in the stomach. And we can quantify the area of the damage. And that's what's shown here. It's we call it a gastric damage score. You can see with naproxen, increasing doses, increasing damage. ATB346, nothing. Nothing macroscopically, nothing histologically, but still getting good suppression of prostaglandin synthesis in the stomach. So we did these studies, and I asked my technician, give the highest amount of this drug you can physically stick through a syringe into an animal, and it was that much. And you can see, we did see a small amount of damage now with ATB346, but much more with naproxen, and actually that level of damage is equivalent to 1/90th the dose of naproxen. So in rats, we see a very good, in healthy rats, we see a very good safety margin. We did a lot of other studies in animals to more closely mimic the human situation. It's sick people generally who are taking NSAIDs, and they have various other comorbidities. So we modeled that. We did a lot of models in animals of comorbidities, and they do have increased susceptibility to GI damage, but the ATB346 still perform very well. So that's the animal data. Now we get to the more interesting part of things, which is the human side of things. So we wanted to we wanted to get this drug into humans. I thought I had a couple more efficacy or toxicity studies there, but they might show up out of order, or I didn't put them in. So we'll go ahead. So this takes us into where we started to do human studies, which was about 2 years ago we started it. And for those of you who may not have ever been involved in these types of trials, I'm just going to take you through the nuts and bolts of it. So when you first start doing human studies of a new drug, you have to, safety is everything. That's the primary objective, is the drug going to be safe to test in people? So the typical way it's done is you start out with single ascending doses. In our case, we had cohorts of 8 people, 6 were on drug, 2 on placebo. We were blind, of course. And you start at a very low dose. So our anticipated dose was 750 milligrams twice daily. I'll come back to that. But we started out at 25 milligrams. So these people take the drug, they're monitored for a week, you have a safety committee meeting, and you decide, yeah, it's safe, let's go to the next dose. And that's how it progresses. 25 to 75, blah, 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 all the way up to 2,000 milligrams single dose. And then we also did a fed versus fasted studying at the 750 single dose. So lots of data generated, things look good, no problems. And then you graduate to the multiple ascending dose phase, where as it suggests, you give the drug to these subjects every day for 2 weeks and monitor them for a couple of weeks after that. And again, you start at a low dose. So again, our target dose was 750 twice daily, but we started at 250 and then went to 750 once daily and eventually to 750 twice daily. The rationale behind that was that we were thinking our drug would be equi-effective to naproxen at the same molar dose, and the dose for naproxen, standard dose, is 500 milligrams twice daily, so that's where we came up with a 750 milligrams twice daily. Now, GI safety is our main interest. We couldn't study that in these healthy subjects, but we did get data on blood pressure as a readout for cardiovascular safety, and that was promising. Black is the placebo group, and these are the different doses. This is 750 twice daily. No significant change in blood pressure. It was measured a bazillion times a day on these people for 14 days and afterwards, and not any hint of increased blood pressure. So that's encouraging at quite high doses. So in the phase 1 trial, doses from 25 to 2,000 milligrams single dose were safe and well tolerated. 250 milligrams for 14 days, safe and well tolerated. 750 milligrams once daily, safe and well tolerated. And then we got a nasty surprise. 750, our target dose, 750 twice daily for 14 days. On day 13, in one subject, second last subject, we had a serious adverse event. He actually had, let me take that back a step. It wasn't a serious adverse event initially. He had an adverse event. He didn't feel well. He had bad abdominal cramps, and he didn't want to take the drug anymore. So that was an adverse event. That in itself isn't that big a deal. They happen all the time. A few weeks later, I was in Brazil. A few weeks later, that's not actually pertinent to the story, but that just happens to be where I was when I got the news. This fellow went to an emergency room of a hospital. That made it a serious adverse event. And that's not good, because that's something that as a company we have to press release. And it's way worse than getting paper ejected. Because our stock plummeted 80% in about 2 hours. It actually plummeted, I think, 95, 96% eventually. We were down to 2 cents a share. And it has, and that's 2 years ago, and it still has not recovered fully. We've had some recovery, but not fully. And we had to go back and figure out what we're going to do, because raising money at 2 cents a share is not going to happen. And so we had to take some drastic moves within the company, but we also had to figure out what was going on. So it turns out that this fellow, we had some bad luck. So these are healthy volunteer studies. The volunteers are paid to be in the studies. I don't know if that's the case in the States or not, if you can pay volunteers for studies. In some countries you can't, but in Canada you can. And it turned out that subject 357 had lied about not having a previous history of hepatitis. He did have a previous hepatitis, but it took 3 interviews with him before he finally admitted that. So he shouldn't have been in the study in the first place. Secondly, he had a gallstone lodged in his bile duct, which was certainly pre-existing before the trial started and would have contributed to the raised liver enzymes, which was the first signal that there was any problem. He also had an active case of athlete's foot that was detected after he was in the trial, and he was found accessing a drug that he was not allowed to bring into the facility, so he was a nightmare. But the problem here is, I think we're dealing with professional volunteers. These are people who get paid for these studies and will lie to get into a study, and unfortunately that just goes with the territory. We did have a second person with raised liver enzymes in that last cohort as well. And it occurred a few weeks after finishing our trial, after he'd been in another trial at the same facility. He also had lied about a prior history of hepatitis. So that's just a caution to any of you who go through this. It's not nice and clean like when you order Sprague Dolly rats and things like that. There are these confounding factors, but there are confounding factors in the real world too. So I guess it's actually something we have to live with. So how do we recover from this? Or what do we do subsequently to try to figure out what was going on? So we went back and looked at pharmacokinetics. So this shows plasma naproxen levels in those various single dose studies we did, going from 25 milligrams out to 2,000 milligrams, and you can see it's pretty much a linear relationship. You increase the dose double, you get double the amount of naproxen. That arrow there just shows the 750 milligrams, the subjects who got that once. So you would anticipate that at 1,500 milligrams, you would have a double, right? You give twice the dose, you get double. It's not quite double, but it's in that ballpark. So if you give 1,500 milligrams, you get approximately double the amount of naproxen as if you gave 750. But remember, we had the problem in the group that got 750 twice in a day. So what happens there? You'd expect the same, right? But actually, when you give it twice in a day, you get very different behavior. And it's because the half-life of the drug is longer than we anticipated. So we get 4-fold increase. And within a week, it's up to 10-fold increase. So what about pharmacodynamics? In that phase 1 study, we didn't collect blood for doing any analysis of COX inhibition or anything like that. We did collect blood. And so I suggested, look, let's get as much of that blood as we can. Let's measure the COX activity and see what's happening, see if we can learn anything about our drug. So we had to go back and get informed consent from subjects, and we didn't get it from all, but we got it from enough that we could do some studies. And I'm just going to show you as an example one patient, because it gave us, let me just get past this. So here's an example. This is someone who, a subject rather, 250 milligrams, so quite a low dose. This is their COX activity, thromboxane production, before getting the drug. And this is 1 hour after the drug. So the first thing we found was, wow, we're getting rapid inhibition of COX activity at quite a low dose. We wouldn't have expected at that dose to see so much. And more importantly, or more interesting, out here, this is now 24 hours after that single dose. We're still getting really good suppression. So when we think about naproxen being a twice-a-day drug, we wouldn't expect that based on naproxen behavior. So ATB 346 is different from naproxen. Let me just go back to those other slides now. So that's just to say that the anti-inflammatory effects of NSAIDs are directly related to COX activity, and that's why measuring COX activity is a useful surrogate, and that's what we did, sorry. So just carry forward from there. So it seemed that our drug might be more potent and more long-acting than we had anticipated. Here's another slide. This happens to be, we got samples from 4 of the subjects who were on very low dose, 75 milligrams, the equivalent of 1/10 the dose someone would take of naproxen twice daily, still getting pretty impressive inhibition of COX activity. You certainly wouldn't see that with 50 milligrams of naproxen. So the phase 1 trial did teach us some things. It was painful, but it did teach us some things. We're seeing enhanced suppression of COX activity versus naproxen, and we're seeing a longer duration of suppression of COX activity. So maybe, if nothing else, one advantage we have is that instead of a patient having to swallow 4 of those a day, they only got to take a half of one of those a day. And it's certainly easier for the patient and some advantage over pre-existing drugs. Not what we're targeting, not what we're looking for, but we have to test that. So then came the next challenge. We thought the way we need to test that is we need to test a drug in humans, in patients, and see if it's reducing pain at these lower doses, or at a lower dose. And that's going to be a challenge because we had those problems in phase 1. So we have to go, our FDA is called Health Canada. We had to go to Health Canada, make an application to see could we do a phase 2 trial. And I think we did an intelligent thing. We took a cautious approach. So we wanted to look at reduction of pain in osteoarthritis. We had done 14 days treatment in phase 1, so we thought, let's do 10. Let's be conservative and only expose patients to 10 days of treatment. Let's go with a low dose, only 250 milligrams. This is the molar equivalent to 1/6 the normal dose of naproxen. The endpoint is the WOMAC pain scale that's standard in arthritis trials. We only did 12 patients, so we kept the numbers low, osteoarthritis of the knee. Started that in April 2016 and completed in August, and we did not do it at the CRO we'd used for the phase 1 trial. We did it in Toronto. The first one was done in Montreal. So here's, just to orient you, this is the kind of thing with these studies. So this is the change in WOMAC pain scale. They start out at 0. The patient does a little scratch on a line of how much pain they have, and this can be quantified. Our study was to measure it the day before they started the trial, when they're in a flare, then on day 4 and day 10. Now, just to orient you, naproxen and celecoxib, mountains of data using exactly this method. The average reduction they both produce is 4, a 4-unit reduction. Placebo produces about a 1.9 unit reduction. So that's what we were hoping we'd get with our drug, that we'd get equivalency to what's been published. This is what we actually got. On day 4, we were around 4. We had already achieved what celecoxib and naproxen produce, and by day 10, we were down to double that. So it's an open label trial, all the caveats and everything, but we were certainly very happy to see these data and to get a clear signal that this dose that's 1/6 the normal dose of naproxen is very potent, and it's a once day. These are just some other endpoints, standard ones, stiffness. And the top panel, again, the green bars pretty much represents what one sees with naproxen and celecoxib in a 10 or 14 day study. And again, we're getting enhanced activity after 10 days of treatment. This just shows the readout that we're actually blocking COX activity, again, in vivo with this low dose. Within a day, we're getting profound suppression of COX activity, and it's maintained. The levels of naproxen in the blood are nowhere near sufficient to produce that effect. They're very low levels of naproxen produced. So naproxen is not the what's producing the anti-inflammatory effect. It's ATP346 or an immediate precursor. Going back to the issue we had in phase 1, that's the plasma, using naproxen as a surrogate, because it's all we have, that's the plasma levels of naproxen in that phase 2 study that produced a good analgesic effect. These are the plasma levels of naproxen in the guy who had the problem. So you can see, and in others in that group. So the problem was the twice daily dosing. They were getting accumulation of the drug, and there were just huge levels of drugs, and that's what we think triggered the raised liver enzymes in those subjects, although they wasn't helped by having a gallstone. So next steps, we are proceeding now. We just did a press release this morning, actually, that we're proceeding, starting in July, to do what's really the most important study in terms of what we're promising, what we were trying to accomplish at the beginning was to have a more GI-safe compound. So we're doing a GI endoscopic study, 120 healthy volunteers. It's in volunteers. I can discuss later if you like. It's in volunteers rather than patients for good reasons, sound reasons, and our clinical advisors supported that. We think if it's successful, that will put us in a position where we can start to talk to companies that may be interested in licensing the product. We're also planning to do, though, a dose-ranging placebo-controlled efficacy study, a bigger study, looking at, with 250 being the highest dose we look at. We're going to look at lower doses and see if we have effectiveness with lower doses. So just to summarize, what we found is linking an H2S releasing moiety to an NSAID in animals substantially reduces the GI toxicity. In humans and animals, it increases the anti-inflammatory potency, but this was seen more clearly in human studies. Also seen in human studies is the increased duration of activity, which we see as a positive of the simplicity of once daily dosing. And these key observations I've reporting to you for this drug ATP346, we've seen with other compounds that we've been developing. We've made many, and at least in the animal studies, we're getting consistency in terms of increased potency and activity. So with that, I'd just like to thank you very much for your attention and just acknowledge my collaborators in various parts of the world who've contributed significantly to these studies. Thanks very much.
And if you could, sir, repeat the question afterwards.
Sure.
What is the longest duration that you follow the patients on ATP346?
14 days. 14 days treatment period followed for another 2 weeks.
You've not shown a change in blood pressure.
No.
But the concern is these increases in myocardial infarction and coronary disease that have been reported with the other agents after a long period of
Right.
use. So I think it's really crucial for you to show that there's no increase in myocardial infarct rates, stroke rates, and so on, despite the fact that blood pressure.
Yep. Yep.
That's really the thing that's killed things like dioxins and these other agents.
Agreed. Yep. Sorry, I didn't repeat the question, but the question was about the long-term significant cardiovascular events that can occur, that have occurred with other NSAIDs, and it is something that definitely will have to be addressed in the future. Yes.
Yes. My question is, at the beginning you started out by talking about hydrogen sulfide and how it was used as a substitute for oxygen, using ATP. So can you tell us a little bit about how hydrogen oxide doesn't do anything in terms of ATP production in mitochondria? Please repeat the question.
So the question is with respect to H2S and its ability to promote ATP production in mitochondria. So I can't tell you much more than I already told you because it's not my field, but there are lots of papers on it, and the Nature Review Drug Discovery paper that I cited there would be one to start with if you want to look further. But certainly the mitochondria can use, particularly in circumstances of anoxia and hypoxia, will use hydrogen sulfide to generate ATP in the electron transfer chains.
Sir, I had a question. Did you look at any markers of hydrogen
Yeah, so the question is essentially, did we measure any H2S or indications of H2S levels in the patients? No, but it's extremely difficult. It's very hard to measure plasma levels of H2S. The methods are getting better. We have, in animal studies, we have more clear evidence, and we can show dose responses and that kind of thing. I can tell you that, again, in animal studies, this is where I thought I had some additional slides. If we substitute the sulfur in ATB346 for oxygen, it's not a GI safe compound anymore. It definitely requires that sulfur for the action.
Dr. Wallace, Abhida, from Harvard. I'm trying to understand the mechanism here. So when you found that naproxen and prodrug, there definitely is a difference. So have you done the control with just the prodrug? I'm trying to understand if H2S
Yeah. So the question is, we have a combination drug of naproxen with an H2S releasing moiety. What would happen if we just gave the H2S releasing moiety? So we've done it in animals. It does not give any protective effect. Even if we give 4 times as much of the H2S releasing moiety, it doesn't give a protective effect. But other H2S donors, we have been able to show a protective effect. That one, and we've done it with several of these different combination drugs we've done. No, it's not sufficient. We do know, because we have measured H2S release from the drug, that you get greater H2S release from the combined drug than from the H2S releasing moiety alone.
Walt? Walt?
Oh, no, that's okay, my question.
Yes, sir.
I have one question. Is there any difference between female and the male in the animal experiment, also in the documented?
The animal studies, I think, were all male. Male-female differences in the responses. Animal studies were all done in males, which is not good, sorry. No discernible differences in the human studies between males and females. Nothing that jumped out. And we looked, but no, there was no apparent difference.
Take your time, take one more question. Yes.
Jay? Hi.
John, can you say anything more about the mechanism of H2S action in affecting the GI? NSAIDs have multiple mechanisms of toxicity. Do you know what the target is of H2S?
So the question is about what's the mechanism of action of the H2S in protecting against NSAID-induced GI damage. The short answer is no, I can't say, this is the reason. But we do see several effects. We see increased blood flow, which is always good in the gut. We see there's increased bicarbonate secretion stimulated by H2S. We actually see with this drug something that could be essential for the small bowel protection in animals at least. So when you give naproxen or most NSAIDs, they undergo intrahepatic recirculation, and that's a big part of the GI damaging effect. When we give ATB346, we don't see any intrahepatic recirculation of the ATB or of the naproxen. So it's curious, and we've seen it with some other compounds as well.
Being considerate of time, I want to end the questions here. We can have further questions during the break, which we're about to take now. Thanks to Dr. Wallace.
Thank you.